Okay, so for this audio lecture, we'll be looking at chapter 14, which has an introduction to immunology, and then it has information on the nonspecific defenses being part of the innate system. So if you've taken anatomy and physiology, you would have already had an overview of the immune system. So this maybe adds in a little more detail on it. But this in no way competes with a full semester immunology course. So again, this is just meant to give you an overview of how the host body defends itself against a pathogen, foreign material. But we also want to keep in mind that these processes are also present when when a human is removing damaged cells in states where there's an imbalance in homeostasis, where there may be dead cells that need to be removed. So we're going to be talking about, because it's microbiology, with the focus of the response to the pathogen. But again, if you were to take an immunology course or even another upper level bio course, you would talk about the immune response to self, so to damaged tissue that's your own. And you might also talk about like transplants and all these other aspects. But again, our focus is going to be on the pathogens. So looking at an overview of the host defense of the immune response, it can get broken into two different branches, the innate or nonspecific, and then we also have our acquired specific defense, sometimes referred to as adaptive. So I tend to refer to them as innate and adaptive just because in grad school, I worked in an immunology lab, and that's where I got my PhD in immunology, and we always called innate and adaptive. As far as test situation, if I ask you a question, an open-ended question, if you use innate and nonspecific, that's fine. So I will accept those interchangeably, just like with adaptive, use acquired or specific, those can be used interchangeably. With the innate, there's two lines of defense. Our first line of defense, which is the physical and chemical barriers, genetic components. So these are like what you're born with in the anatomy of different structures. And then we have our second line defense, and our second line defense is what's the cellular response. So what are the cells doing? What chemicals are they producing in response to a pathogen, to damaged cells? And especially when the first line of defense has been broken. Okay. And then with our adaptive, this is what we would call our third line of defense. So this is what our lymphocytes, our B cells and T cells are doing. B cells produce antibodies. And so again, it's all that the response of these B cells and T cells and their products and what effect they have on the immune system. So looking at the first line of defense, this really is barriers. So we have, the physical barriers because of the anatomy, but there can also be chemical barriers in there also. And then we can also have genetic factors. So there's certain, differences because of our genes, because of what we've inherited in our immune response, okay, that play a role in how these first line of defenses work. Okay, when we look at physical barriers, our skin is going to be our biggest physical barrier between the outside world and us, inside, protecting us. And so again, when there's a break in the skin, there's a break in the first line of defense, and that requires the second line of defense to kind of step in. We also have mucosal layers, cilia, and different secretions, all that aid in protecting and creating this first line of defense. So if we think about, our respiratory tract, we have all this mucus production that helps to trap microbes so that they can't actually, damage and attach to our respiratory system. Okay, obviously the pathogens have their own tricks, and we talked about that in chapter 13, looking at microbe host interactions. So, viruses and bacteria can change our mucus production so that they're more likely to be able to get in. Okay, and then with cilia, those are that hair-like kind of structures that beat, and so again, we have cilia in our respiratory tract to help move that mucus that could have microbes in it out of the respiratory system. Now, the basic premise of the immune system is being able to recognize self and non-self. So our white blood cells, our leukocytes, are able to distinguish self from non-self. So they have receptors on their surface that allows them to say, oh, that's me. Nope, that's not. So there's receptors that are specific to pathogens. There's receptors specific to damaged cells. And then you have barcodes on your own cells, like your liver cells, your skin cells. And so again, white blood cells, leukocytes, and lymphocytes would be able to distinguish between self and non-self. And again, it's through these surface markers that they do that. So looking more specifically at the innate response, looking at the cells that are involved, so these would be part of our second line of defenses. These are the players, these are the actors, these are what are going to contribute to inflammation, phagocytosis, complement, and also interferon production. So again, you might have covered this in another class like A&P, but leukocytes is another word for white blood cells. They fall into two different categories: granular cytes. Granular cytes have granules in them, so if you stain them, they have dots. So they have these large dots because the granules will take up the different stain. The granular cytes come in three different types. Neutrophils. Neutrophils have a neutral pH in their granules, hence the name. Basophils. Basophils have a basic pH. And you can see with basophils, they have these dark purple granules. And then we have xenophils. Xenophils have an acidic pH. It's not in their name, but it's the other one. And so their granules stain red because of those acidic components in their granules. We then have agranulocytes, so without granules. So they do have granules, but they're very small, so their cytoplasm has a more even appearance to it. It doesn't have all the little dots when you look at it underneath the microscope. This includes monocytes. Monocytes become macrophages, and they're part of the innate response. But then we also have the lymphocytes being part of the adaptive response, and they fall into the T and B cell categories. So this gives you a more realistic, like, what do they look like underneath the microscope? If you have me for A&P2 in lab, I'd make you look under the microscopes. I'd make you make a dichotomous key so you could identify them all. And on part of the lab practical, there'd be like 20 different images, and you'd have to identify what they were. So I love microscopes, and I love the different cells and the structural differences between them. Xenophils are probably my favorite ones because, as you can see, that middle image, they have that really nice red coloring to them, which make them really distinct and easy to identify when you do a Wright stain. So WRIGHT staining. It's a special combination of dyes, and again, it can distinguish between the granules, whether the granules are acidic, basic, or both being neutral. And so again, if you have acidic, you're going to have red. That would be in a xenophil. If it's basic, that's down at the bottom where you have the dark purple, and that would be a basophil. And then lastly, neutrophils. Neutrophils are going to be a lighter purple color. And again, they would be a neutral pH. With the neutrophil, the way to tell is the nucleus. So it's multi-lobed. So that's one of the ways to distinguish. So all of these cells can act as phagocytes, so they can engulf. Neutrophils are known more so for that because neutrophils can engulf bacteria, so really important for microbiology. Xenophils are known for producing chemicals that actually will paralyze parasitic worms. So not so much, it used to be believed that they would actually engulf the parasitic worms, and it was this David and Goliath thing. But what we know now is that they actually produce chemicals, and those chemicals will actually paralyze the worms, so the worms can't actually attach to the intestines, and then they are shed. Basophils are known for producing histamine, so they release histamine during the inflammation, during the immune response. And then with our agranulocytes, if you look at them with the lymphocytes, the T cells and B cells, that's what would be up top. So they actually have what's known as a cytoplasmic brim. So this very small amount of cytoplasmic material. Where the monocytes will actually have a kind of kidney-shaped nucleus, which is kind of characteristic of it. And again, with agranulocytes, this is where we would see this like lack of the granules, the dots. You can see how their cytoplasm is clear of those granules. And then with macrophages, we just want to keep in mind that macrophages are found in different locations. So we can have macrophages that are free in circulation in the blood, but then we can also have ones in special sites like in the lung, in the liver, and also in the skin. And they have, at least for the liver and the skin, they're named after the scientist who found them. So Kupffer cells are if you ever hear about them, they are actually our macrophages. They're just in the liver. And then Langerhans cells are found in the skin. And so again, just a macrophage that's a resident there. So your resident macrophages, the ones in the lungs don't have a special name. They're just alveoli macrophages. And so the resident macrophages are a little bit more knowledgeable, so mature. They have learned about what pathogens are normal for being in those locations, opposed to your free macrophages. So when we look at our blood cells, they all originate from a stem cell. So we have our hematopoietic stem cell that both are innate and adaptive cells originating, and even our red blood cells also. We have two different lineages, if you will. So we have a lymphoid progenitor, and then we also have a myeloid progenitor. Okay, and so when we look at, if you look at the yellow box there, we have our granulocytes originating from that myeloid progenitor. For agranulocytes, our T cell and B cell are going to originate from the lymphoid progenitor, but the monocyte's actually a myeloid, so it originates from the myeloid. Okay, from the myeloid lineage, we also have our platelets and red blood cells originating. We also have a special type of cell called mast cell. Now, mast cells are similar to basophils, that they release high amounts of histamine, but they're found in the skin. Okay, so I always kind of think of them as basophils in the skin. The lineage is, they're not the same because their lineage is differentiated at a higher level at the progenitor stage. Okay, and then we also have natural killer cells. Natural killer cells actually come from the lymphoid lineage. Natural killer cells are really important in regulating T cell response, but in recognition of damaged cells that are our own. So really important in cancer therapies and looking at how to modulate and change the natural killer response. Okay. And then keep in mind that, again, our macrophages and dendritic cells come from the monocyte. So the monocyte then differentiates into a macrophage, and that could either be free or a resident, and then also a dendritic cell. So you definitely should know the progenitors, the lymphoid versus myeloid progenitors, and you should know the hematopoietic stem cell and that they originate, that it originates from. It's not that you have to memorize and fill it. You're not going to get this figure on the exam and you have to fill it all in. But again, this figure kind of helps to show the lineage of them. So looking at the second line of defense is these cells that are going to end up having a response to a pathogen or damaged tissue. And so those responses can come in different processes, cellular processes. We do want to keep in mind that these processes, the second line of defense of the innate, really is important for the third line of defense for adaptive. And so we talk about innate versus adaptive and how there are these two branches, but really there's a lot of interconnection, and it's these processes that really connect them. So if you have inflammation, part of inflammation is so that you can get your adaptive response going. Part of having phagocytosis is so you can get the adaptive response going. A complement actually is interferon. So going to interferons are being produced, and your adaptive cells actually sense the interferons and become activated because of them, or not activated, depending upon what interferon. With complement, a lot of times this is kind of put into its own category because it really branches between innate and adaptive. So there's an innate component because you're born with, your genes that code for complement proteins. It can get activated without having adaptive involved, but one of the pathways, the classical pathway, requires an antibody to be there, and that really means that adaptive had to have been activated and utilized before in order to get complement. So again, though these are being put in the innate category in the second line of defense, keep in mind that there is a crossover into the third line of defense. So looking at inflammation, this is whenever we have some kind of trauma. So that could be some kind of injury, whether we got a paper cut, whether we stepped on the rusty nail, or whether it's even something like you banged your toe, and maybe there's no break in the skin, but there's going to be tissue damage because of that. So you would have like a bruised toe, and you need to repair those cells in there. Okay, the classic or cardinal signs and symptoms of inflammation are redness, warmth, swelling, pain, and sometimes loss of function. Okay, so what's the benefit of inflammation? It's that repair process, and it's also, if there's a break in the skin, it's also making sure that pathogens aren't present and removing them, right? So we want the benefits, returning the tissue back to its normal functions so that it can function, the way it's supposed to, its normal physiology. Okay, so we're going to go on a magic school bus, and we're going to travel to the site of inflammation. So here we have our rusty nail that has microbes all over it, so you can see all the bacteria that's been introduced into the wound because we have a break in the skin. You can see how we have mast cells that are present, and so those mast cells can release chemicals, histamine, you should know histamine, not just chemical mediators. And so initially you're going to get a vasoconstriction of the blood vessels nearby because there's been damage potentially to them, and so we want to stop blood flow to the area. And again, this is just a short, immediate response. Once we get this histamine release, we're actually going to cause dilation. So you can see in panel 2 how that blood vessel now has dilated. So keep in mind, vasoconstriction, blood vessel becomes smaller, slower blood flow to no blood flow, depending upon how constricted. And then with vasodilation, opening up of the blood vessel, so the diameter is going to be bigger, so we get more blood flow to the area. Okay, with that blood flow to the area, white blood cells can come into the location. So we can have neutrophils and other immune cells, eventually lymphocytes, coming into the location so that they can undergo phagocytosis with our bacteria. Okay, and we'll talk more about the process of phagocytosis, but we can see how our neutrophils are in there. Our neutrophils, our bacteria slayers, are going to go and engulf our bacteria. We also have a clot that has formed so that we don't lose fluid or blood in that area. Okay. As inflammation progresses, where you're going to have some swelling in the area or edema, because of that vasodilation, plasma and fluid from the blood can leak into that area. We also have all these cells that have entered into the area that are metabolically active. So they are, undergoing phagocytosis, clearing out the bacteria, the damaged cells, and then basically regurgitating them out. So that ends up leading to this formation of pus in the area because it's all that debris that these neutrophils have processed and released. Okay, at this point the clot has begun to contract, so we're going to get a formation of the scab. Okay, and then eventually we recruit in lymphocytes to make sure that all the bacteria is gone. Usually the lymphocytes that are specific to that damaged cell or for that bacteria would have been recruited into that area. At this point, we've had a chance for mitosis to occur of skin cells and also of the endothelial cells in the blood vessel, so that we have repair of those different structures. That repair process can leave a scar due to pigment changes in that newness, if you will, of the skin cells that are present there at the site. But again, we have the lymphocytes that have been recruited in. We can have antibodies being produced also. And we can also get additional macrophages, because we would have our resident macrophages, but some of the free macrophages can come in and also help with phagocytosis, clearing out of any additional bacteria that might be present. So I did leave you a little slide for taking your notes on inflammation. So if you do print out the notes, you can add in those pieces, right? So one of the things we do want to think about with inflammation is, how does this correlate to those cardinal signs? So we have redness. So what's causing that redness, right? So we have increased blood flow to the area, and that means more red blood cells. So that's going to be some of the reason for the redness. Okay, because we have that vasodilation, so increased blood flow. We have heat, right? So some of that can be because of that increased blood flow, right? So one of the ways we warm our bodies by increasing blood flow, because our blood retains heat. But we also have all these metabolically active cells, and what I mean is, when a immune cell, when a phagocyte is undergoing phagocytosis, there's all these chemical processes that have to happen. So they're really active, and each one of those processes, a little bit of heat is being lost, a little bit of energy is being lost as heat. And when you put that together with thousands of cells in that location, that heat has this additive effect. So that in a site where you have inflammation, where you have these phagocytes, if you touch it, it would be warm. We already talked about the swelling, right, the edema, because of all of the additional cells in that area, also the fluid, the plasma from the blood that's moving into that area, so that would cause some of that edema. We also have in there pain, right, and so we have receptors all over our skin, all over our body, and those receptors can get signaled, so we perceive that as pain. And then with loss of function, that ties in with the nerves too, because if the nerves aren't signaling, or they are signaling in pain, one of our things we do is we stop using that area, right? So don't use it so you don't hurt it again, let it repair, okay? So sometimes because of those nerve impulses, we can lose the ability to move that area. An important process with inflammation, we said that these cells get into the site where we have inflammation, and it's through a process of diapedesis and chemotaxis. So chemotaxis is where we have these chemical signals, so like the histamine, but we would also have other factors being produced by the tissues. So the tissues would produce factors that would get into the blood, and so immune cells would be able to sense those, and then would be drawn to the location where it's being expressed at higher concentrations. So this is where we would have like positive chemotaxis movement to an area or to the tissue that's damaged, because that's where the site where the chemokines and these factors are being produced. In order for the immune cell to get out of the blood, and into the tissue, it has to undergo a process called diapedesis, also sometimes referred to as margination. So textbooks have kind of dropped off diapedesis and just said margination. Margination is also a process in cancer cells. And margination is when a cell is moving in the blood and then it starts to roll and attach to the endothelial layer and slow down. Diapedesis, that's squeezing through, as you can see here, squeezing through the space between those endothelial cells. And because we have dilation, there's a little bit more separation of that, of those endothelial cells, so that neutrophils, other immune cells can squeeze through. Keep in mind that red blood cells are rigid, they can't squeeze through, so they'll stay in the blood vessel. But again, immune cells are able to kind of change their shape and squeeze through the process of diapedesis. And then I left a space for any additional notes. Looking at the process of phagocytosis, this is a way in which material can be cleared out. So again, pathogens, microbes, also any kind of matter that shouldn't be there, like pollen and such, but also for injured or dead cells too. So the phagocytes, the cells that undergo phagocytosis, and we've talked about how neutrophils can do this, but your macrophages, your dendritic cells can do this also. B cells can do it. They don't do it very well. They're known for producing antibodies, but so it's not just one type of cell that is a phagocyte. A phagocyte, that's a general term for cells who undergo phagocytosis. And what they're going to do is ingest the material and then they break it down and eliminate it. Okay. With this process, again, this interacts with the adaptive, and so there's a lot of information, antigens that can be presented to the adaptive system from here. So when the material is eliminated, it's not necessarily just spit back out. It can be loaded onto receptors that allow for the phagocytes to talk to T cells and B cells, so that if they recognize that antigen, those T cells and B cells would become activated. So again, there's a lot of cross communication between innate and adaptive. So this outlines the different steps of phagocytosis. So it's initiated by chemotaxis, but then we need to have direct interactions between the receptors and ligands, the receptors being PAMPs, pathogen-associated molecular patterns. Just be aware that some textbooks are switching to MAMPs, so microbe-associated molecular patterns. There's actually plant-associated molecular patterns in botany, not specifically related to how your immune system functions. And so the botanist kind of won, and so we've kind of updated it to be microbe-associated molecular patterns, and it's just one of those things I haven't fully converted over yet. I will at some point, but PAMPs, MAMPs would be microbes. There's also DAMPs, damage-associated molecular patterns, that are found on damaged or dead cells. Okay. So these molecular patterns bind to receptors, PRRs, pattern recognition receptors. So an example of a pattern recognition receptor is a toll-like receptor, but there's also nod-like receptors, and there's all these different pattern recognition receptors, okay? Once we get that direct interaction between the ligand and the receptor, that initiates ingestion, internalizing of that material, of that pathogen, and then we put that into a vesicle, into a phagosome. So a phagosome is a vesicle that contains the pathogen. That's going to end up fusing with a lysosome. So the lysosome contains digestive enzymes. When those fuse together, we form a phagolysosome. So we're just kind of like put the words together. So that phagolysosome is going to contain the pathogen and the digestive enzymes so that they can mix together. The digestive enzymes, there's also a drop in the pH. That leads to the pathogen being destroyed. It gets broken down into little pieces, just like when you digest your food. And then that vesicle goes through exocytosis, so it secretes out that waste material, keeping in mind that there's also an opportunity to load this onto receptors so that lymphocytes could interact with it. So this just shows you this in a figure, in a visual. So again, we're going to have our phagocyte drawn to location because of chemical signals, chemotaxis. And then we get direct interaction between the PAMPs, the molecular patterns that are on the pathogen, and the receptors that are on our phagocyte. Okay, so we have our pattern recognition receptors. That is going to lead to engulfment, so that the pathogen is enclosed into a vesicle. So you can see in step 4 how we have our phagosome. That phagosome is going to bind with the lysosome that has the digestive enzyme. So that's going to form the phagolysosome. And then that's the digestive enzymes with the drop in the pH is going to lead to the bacteria, to the pathogen being destroyed. You can see how we have different types of enzymes present. Some of them are going to break down the proteins, some are going to break down nucleic acids, some are going to break down the lipids, so that we get effective breaking down into these little residues. So obviously the microbe is no longer alive, it can no longer reproduce. And then we have our exocytosis, where the vesicle will move to the membrane and then will undergo exocytosis, secreting out that material that's in there. And again, there would be receptors that are on that vesicle, allowing for loading of those processed material onto them so that they could be presented, once they got to the surface, to a T cell, to a B cell. So this video outlines the process of phagocytosis. I'll let you watch that on your leisure, just because that running of a video and the capturing of the audio, sometimes it doubles over. But just this gives you a visual of the moving pieces of phagocytosis. So then also with the innate response, we have interferons and complement. So for interferons, this is a way in which cells can communicate to immune cells if they are virally infected or compromised in some way. So, I mean, at the same time, viruses try to downregulate interferon production and other factors like production of receptors on our cells. But again, kind of the interferon becomes a language to immune cells saying something's wrong with me. It also can communicate to nearby cells to take up certain defenses. Okay, there's 3 different types. There's interferon alpha, interferon beta, and interferon gamma. Gamma tends to activate the immune response, so it'll activate lymphocytes and other cells. Interferon beta decreases the response. So interferon beta is actually used in some autoimmune disorders like MS, multiple sclerosis, and so interferon beta is given to those patients because it quiets those immune cells. And then interferon alpha depends upon the cell type, whether it activates or decreases. So here we can see how we have this virally infected cell, and once it senses it's virally infected, it actually will turn on those interferon genes. It will synthesize interferon and then secrete it out. And so this allows neighboring cells to sense the interferon because they have receptors for it, so that they can mount a defense. It also would signal to immune cells. So immune cells could sense that and then respond and help to clear out those infected cells also in that way. For a non-immune cell, what it can do is actually produce enzymes that will help to degrade the virus. It can block the replication, so it can produce all these antiviral proteins that help to fight against infection. All right, so next and lastly, we'll talk about complement pathways. So for complement pathways, they're made up of complement proteins. Complement proteins are found in our plasma, in our blood, so they're in circulation. So right now, your complement pathway is at work. Whenever you brush your teeth, it's at work. And so a really important part of your immune response that we tend not to think about too much. And so there's three pathways for complement. There's the classical pathway, the lectin pathway, and the alternative pathway. So the classical pathway is fast, it's very directed, and very effective. Okay, so it actually gets initiated by an antibody. So an antibody will bind to a microbe. And in binding to the microbe, it allows for a C1 complex to form. And so the C1 complex is a group of complement protein 1. So there's different subunits. They come together, form that complex, and then that causes additional complement proteins to become activated. So with complement proteins, for most of them, they have a inactive form, and then when they become activated, they get cleaved and broken into two subunits. So what happens is your C4 and C2 become activated. They cleave into two subunits, and some of those subunits bind to each other. Other ones go off and cause inflammation and signal to say, "Oops, something's going on here. We've been activated." And so C4b and C2a will bind together, and they activate C3. C3 becomes activated, it breaks into two parts. C3b will bind to that C4b, C2a complex, and then we'll activate C5. C5 becomes activated, and it forms C5b. That then binds to 6, 7, 8, and a bunch of 9s, and that ends up making a membrane attack complex. Okay, so the membrane attack complex gets embedded into, as it's made, is embedded into the microbe. And so it actually creates this pore, this hole, so that whatever's inside of the microbe leaks out. Okay, at the same time, whatever's outside can leak in, and so the microbes can't survive that, and so they end up dying. Okay. Now, there's two other pathways. There's the lectin pathway and alternative pathway. The lectin pathway requires that there's this mannose binding lectin, so it utilizes lectin to bind mannose. Not all microbes have mannose on it, but some do. So it doesn't require an antibody to get started. Instead, you have these lectins that are going to bind to mannose, a carbohydrate on the surface of the bacteria. So it gives you a way to respond if you haven't seen it before. And from that binding, you then get activation of C4 and C2. So you get that same complex of C4b2a forming, and you get, and for some reason this figure has this missing, but then you also get C3 becoming activated, and then you end up getting the membrane attack complex because you get C5 activated, and then 6, 7, 8, and a bunch of 9s binding. Okay. With the alternative pathway, this is a slower pathway and it's less effective because it spontaneously activates. So just keep in mind with classical and lectin, they're activating in response to something. With alternative, it's like, we became activated. Is there something that controls this right now? We don't really understand that, so we say it spontaneously happens. And so if you have activation spontaneously, you don't want to have it happen all the time. So you want it to be a slower process where it's like checkpoint, do we want to move forward? Checkpoint, do we want to move forward? Okay, so with alternative, C3 will spontaneously become activated in the blood. C3b will then bind to the surface of bacteria, of a microbe, right? So you do have to have the microbe present in order for the C3b to bind to it. A little different, so we don't have the C4 and C2 involved, but we have these other factors. So we have factor B or factor D that can bind to the C3b. We also have propidine. Propidine actually stabilizes this whole complex and allows it to kind of anchor to the microbe. We'll get an additional C3 being activated. and then we'll get an activation of C5, okay, and then the membrane attack complex forming. So we have this kind of convergence of C3 becoming activated on all of these pathways. Again, classical requires you to have seen the pathogen before, so you've had to have brushed your teeth before and seen that pathogen. Lectin, the microbe has to have mannose. Not all of them have that. So alternative gives us an alternative way to still activate complement, even though we haven't seen it, and it doesn't have that specific carbohydrate on its surface. In all three of these, we get these subunits that can go off and cause inflammation. Generally, that's your A subunit. And then we also get the formation of the membrane attack complex. Complement pathway has been looked at kind of increasingly with COVID, because there is high activation of complement pathway with COVID. So it can be activated not just because of microbes or bacteria, but also because of viruses. And so in part of the immune response to COVID, C5a is formed. And so that C5a causes additional inflammation, actually can cause the cytokine storm where you get upregulation. So especially COVID patients with severe acute respiratory disorder syndrome, that individuals that are going to need to be vented, they have upregulated or increased levels of C5a present in their serum when in one of the studies, and I can post the study. So there are actually C5a receptors on your immune cells, and so one of the drugs that are in clinical studies in Europe and France is looking at, and it just entered, I believe, in the US. It actually blocks that receptor, so the immune cells can't sense that there's all the C5a out there. So then you don't get cytokine storm and you don't get that damage to the lung and such. Okay. That drug was originally designed for cancer therapy, to actually kind of activate immune cells, to retrain them, if you will, so that they would respond. But it's showing potentials in some of the initial studies, and definitely in the in vitro models where they've taken mice that have high amounts of C5a and actually blocks the response, so that the mice don't get any of this damage and cytokine upregulation. So again, just kind of linking this back to one of the figures in the textbook. Again, we have initiation. This is looking at the classical pathway, because we see an antibody binding to the surface to an antigen that's on the surface of the microbe, and then our C1 complex coming in. We then get amplification of that signal, because we're going to get activation of C4 and C2. So we have C4b2a now present, and that's going to activate C3. So it actually is called the C3 convertase. So C3 convertase converts C3 to its active form. So we now have C3b. C3b joins that complex. So we have C4b2a 3b. This is recognized as C5 convertase because it's going to convert, it's going to activate C5. So C5 gets activated. So now we have C5b. It then starts the process of building our membrane attack complex. So C6, 7, 8 will bind, and then we have all these C9s that end up forming a ring. And so we end up getting all these little pores forming. So you can see that it's not that we get one of these formed. We're going to end up having in the area there's a high number of them so that the bacteria lose its ability. So we get membrane attack. So we'll definitely go over complement just because I think it's a difficult process to kind of wrap your head around, because there's all these little pieces, all these complement proteins involved, and their subunits. So it's a little bit easier to kind of whiteboard it out. It seems like it doesn't matter how many times you look at the figure, still like, oh, I don't see it. But once you kind of whiteboard it out, it becomes a little bit easier to understand. So as always, if you have any questions, please do not hesitate to contact me.